The overall goal of the following experiment is to preserve the natural distribution of glycosylation of secreted mucus in tissue samples. This is achieved by first embedding the tissues in optimal cutting temperature medium, or OCT, to minimize the tissue processing. And then, to allow detection of glycan structures, mucins, and mucus, the sections are incubated with lectins, antibodies, and histochemical stains. Lectin binding can be further challenged by competitive inhibition or enzymatic cleavage of glycan-epitopes to confirm lectin binding specificity. Ultimately, the preservation of mucus in frozen tissue samples can be compared to the preservation of mucus in paraffin-embedded samples by histochemical analysis.
The main advantages of using this technique over existing methods, such as embedding tissue in paraffin and fixing with Gagneux solution, are that this method does not require the use of specialized fixatives and allows the utilization of frozen tissues that may already be existing in the lab.
To begin, allow the snap frozen tissue to warm up to -20 degrees Celsius by placing it in a cryomicrotome chamber. Meanwhile, create a freezing bath by adding 2-methylbutane to a shallow styrofoam box and then adding dry ice to the box. Next, add just enough OCT to cover the bottom of a peel-away freezing mold and then place the tissue in the mold. Make sure that the tissue rests on the bottom of the mold in the desired orientation. Cover the tissue with more OCT and then place the mold in the freezing bath. The OCT compound will turn white as the tissue freezes.